Rodney Samaco, and I'm going to talk about why models matter, because I think for those in the room, I believe 50% of you had attended the 2023 conference, right, for the CTNNB1 Connect and Cure conference in New Jersey, and I believe that many of those from Med13L are new. And so when you're hearing today, what I want you to walk away from the talks later this afternoon is basically, why do scientists study animal models, cell models, et cetera? And what is the motivation for it? And where do you fit in? Okay, so a bit about myself, how I got here. So basically, I had an academic lab. I'm a reformed academic, I would say, focused on rare neurological conditions, primarily neurodevelopmental disorders and rare epilepsies, working on mouse models and rat models, looking at biomarkers and outcome measures, preclinical work, right? So I've worked on a couple of different assets, developing gene therapy for a condition called CDKL5 deficiency disorder, as well as an ASO for Angelman syndrome. And that was great. But the reason why I moved forward was because of working in the greater space of advocacy and policy. And so then I joined a nonprofit organization in Washington, D.C. called AUCD, that's the Association of University Centers on Disabilities, where I worked on strategy and business development, and then more recently left AUCD and started my own consulting venture called Rare Collective Strategies, based in Chicago, Illinois. And so the motivation I want you to think about when you're hearing talks later is, when scientists and communities are working on various models, especially rare genetic conditions, they're asking themselves, can we identify something that's reproducible and reliable? And can they be preclinical surrogate endpoints in basically, I say rodent models, but it could also be fly models, as Dr. Chow will speak about later. They could also be cell models, as Dr. Jacobs will speak about it later as well. And so outcome measures and biomarkers you've heard about briefly, but the most important here in orange, proof of concept, right? So we want proof of concept studies in model organisms before we go into people, because we want to ensure that things are safe and they're efficacious. And then we also want to ask the question, do they track with the experimental therapeutic manipulation? Meaning that if you expect something to be upregulated or downregulated, does the drug or genetic therapy actually reverse that to make it neurotypical? Finally, we want to ask questions such as the onset and progression of disease severity, and the aspect of overcoming, I would say, challenges to team science, those conceptual and technical issues, as well as human capital. I think many people within this space, especially the rare disease space, understand, that it's sometimes very difficult to find the time to volunteer, to find the time to really make change. But that is a challenge that hopefully all of you in this room, and the reason why you're here today, are working on. So often we ask ourselves as scientists, basic scientists, focusing on your North Star. So what is our North Star in terms of working with animal models? Are we trying to understand the biology, or are we trying to develop a therapy? And so those two things can work in tandem, but sometimes they appear to be isolated. And so stitching together that journey, right? You've gone through the genetic odyssey, right? The diagnostic odyssey, and now you're working as a community, working towards a therapy to develop one. And the researchers who are working on animal models are trying to leverage, whether they be in vivo or in vitro, meaning in life or in a dish, whether they could leverage their models to identify those preclinical biomarkers and outcome measures. So one is the goal, and the other is the motivation. So it's kind of discovery versus hypothesis-related experimental design. And so if you're looking at things such as developmental delay in people, or seizures, EEGs, speech, cognitive, motor, behavioral, you ask yourself, how can we reproduce that in an animal model in something like a dog or a mouse or a rat? And I would say you wouldn't, right? Because you're not trying to say that a mouse is a person. We're not trying to what they call anthropomorphize the model system, but rather we're trying to identify at baseline, how is it when you take a single variable, such as a gene, and modify it, reproducing an effect that could be used for a preclinical study. Again, preclinical studies are needed in order to ensure safety and efficacy before we go into people, and that's the most important. So on this slide is an example. So let's say, for example, you knock out a gene. So when you hear knockout, basically all of the genome in that animal model or cell model is retained, and that single gene is modified. then ask the question, what happens during life, that period of life for that animal? Is it instructive? But then what is the intent, right? What is the intent of the researcher? What is the intent of your hard-earned fundraising dollars going towards and supporting investigators in the lab? So the intent is, what can we learn from the model? Can we use the models as tools to develop potential therapies? But what can we also learn independent of the why, right? We may not understand the mechanism of why a drug is working for many years, but perhaps there might be some reason to screen the animal model and test compounds that already exist and have been approved by the FDA. Okay, so that was like a lot, right, to digest, and I'm trying to go very quickly here. So how does it fit into the journey towards clinical trial readiness? That's what I was asked to speak about, because when you think about preclinical work and clinical work, how are they intertwined? And in fact, they are well wedded together because you need one in order to advance forward to the other. And so for you all, why should you care, right? And who should care is a big question, right? What do you do next? As you walk away from this conference on Saturday, you're going to ask yourself, okay, I've learned a lot of things, I've made a lot of connections, but how do I get involved? You're here today, okay? That's why you're going to be involved. And I'm going to show this slide here, and it's a very busy slide, but basically borrowed from Alison Berendt, who many of you may know or have heard of in the Angelman syndrome community. So she is a force of nature and has been very inspirational for the community in terms of what family organizations can do to basically push the needle forward and ensure success for their community. And they currently have ASOs that are trusted in their community, Angelman syndrome community, as well as other drugs that are coming down the pike. And so this can happen for you as well. I would say in terms of being proactive as a community, one thing that you look at sometimes is this kind of key assets, right? So how are you preparing yourself for clinical trial readiness? So 1 through 5, I'm not going to go through everything here, as I think many of you will be covering this later as the speakers, and Anna already spoke about, you have a disease concept model that's underway, you have the natural history study, et cetera. But looking from 1 through 5, just meaningful readouts, that's important. Preclinical models, so that's work that's ongoing and will be discussed. Those partnerships and collaborations, you're here together as a community already collaborating because of the common clinical overlap and also common interests. And finally, having that proactive and engaged global community will help boost, I would say, attention towards CTNB1 and MED13L. Okay, so community domain expertise and resources are necessary components to find answers quickly and often, but sometimes we don't talk about those resources. And those resources can be either funds or people, right? And I would say all of that works together in order to move the community forward and be that activated energy to find a solution. So here, tell me more, how can I get involved? So all of this here on the right in the green box, all of those different skill sets, many of you already have them, and some of you have been untapped, or maybe are trying to wonder or ask yourselves, like, how do I get involved as you leave on Saturday? So these are things that would be certainly useful for the community. One way to connect, email Med13L. You can see the email address there, volunteer@med13l.org, or scan the QR code and for CTNB1, and you could find ways to connect and cure CTNB1. How's that? All right, so two more slides here. So how community needs and engagement can lead to change. So in 2019, one of the groups that I worked with, the CDKL5 deficiency disorder groups, they hosted a patient-focused drug development meeting to basically bring the FDA representatives to the table and tell them, this is what's important to our community. And so there's a small slide to the very right and top right about what was important to them. And this is a rare epilepsy, severe epilepsy condition. And while epilepsy was the common thing that a lot of physicians and clinicians were looking at, that wasn't the primary goal for the parents. The parents wanted to know, how can I get my kid to talk? How can I get my kid to communicate with me? And so that was really insightful for the FDA because they thought this whole time, okay, well, we're just gonna focus on the seizures. But in fact, that they knew, the parents knew, I will live with this my entire life and my child will as well. I want my child to live a full, independent life and be able to communicate. That was more important to them. So this is something that I think hopefully soon that the CTNB1 and Med13L communities can also do. And that's it. So thank you, everybody. And I ask you that you should be or consider being part of the solution and shape the future for these two communities.